Well, let me first say thank you to the organizers for allowing me to be here. Great conference. I'm going to be, maybe very first thing I should do is just talk about just a brief mention that a bunch of collaborators involved in the work I'm going to tell you about today in US, students and postdocs at Austin, and some from over here in Europe as well. And what I'm going to tell you about is a little bit about visual search and identification in the real world. So looking for specific targets is something that we're just doing all the time. It's just a continuous natural behavior that we're involved in. So it's very fundamental, sort of basic thing that we do. And for the last, 15 or more years now, we've been sort of, actually quite a bit more actually, if you think about where the real beginnings are, but we've been working on trying to develop a sort of a formal theory based on first principles of a visual search for targets, and recently including natural scenes. So I'm going to tell you about that. There's some overlap of this with the very fine talks that we had from Ruth and Felix on Monday, but a little bit different in some ways. I'm going to be presenting quite a bit of specific details. So a bunch of little parts to this talk. I believe that they form sort of a coherent whole. Every one of them is not that hard to follow, but as a whole, there's quite a few pieces, but I think they're coherent, so we'll see how that goes. Stop me if there's things that are unclear. So the fundamental principle that's been sort of guiding our work for some time is what we like to call natural systems analysis, and the fundamental idea is natural selection. Perceptual systems, including their mechanisms for learning and plasticity, they evolved to obtain information about the environment that's relevant for the tasks that a specific organism performs. And different organisms perform different tasks in order to survive and reproduce. That's where they came from. And the consequence of that is that we would, because of natural selection, we're going to expect our perceptual and cognitive systems, including the mechanisms for learning and plasticity, to be matched to what the organism's tasks actually are, and not to arbitrary tasks. So if you take this seriously, you sort of led to something like this as an approach for doing vision science or perception science. First, try to characterize, well, what are those natural tasks? What is it that you do? Then go and actually try to measure and analyze the relevant environmental properties that are in the environments, natural scene statistics, as well as biological constraints in the organism that we sort of know are there, measure and analyze those that are relevant to those tasks. And then determine, and then the next step is sort of kind of more of our thing, and that is try to determine how to use those measured properties to optimally perform whatever that task is. Now, lots of times the tasks are, we start kind of with, if you like, subtasks or constrained tasks, but then slowly over time try to expand them. It's hard to determine what's optimal for many tasks. But anyway, we think of these first three steps as sort of the macro physics of natural tasks. And it'd be relevant for artificial vision systems or perception systems as well as for biological. And we're mostly interested in how the brain works. And so what we do is we use this macro physics of natural tasks, and especially the idea of seeing what you'd want to optimally do with that information, to give us some principled hypotheses about what to look for in the biological systems, either in behavior or in physiology. So it's getting, in some sense, it's getting principled hypotheses here that's the big deal, and then we go in and try to test them. And of course, we don't expect organisms to be optimal, but the idea here really is evolution has had a lot of time to keep pushing the organisms in the direction of doing the right computation. And so it's a good place to start. And then you can say, well, what heuristics as Roland was pointing out, would get close to doing the right thing, right? So it's a good way to get started on that. Okay, so we're not going to go completely general here with huge tasks. So we're not going to talk about as many different tasks as Ruth talked about. I'm just going to focus on this. This is the constraints for what we're going to do here. I'm going to consider the stimuli to be sort of background, the arbitrary background images. They could include natural images. Some known additive target, that's just a target that's added somewhere in the scene. Here you can see there's a little gaborlet or something, but some target that's added at some location in the scene. There's some prior on whether the target's present or absent. There's some prior over the possible locations where the target might be located. And we consider covert and overt search. So covert search means that you just have one essentially fixation to decide whether the target's there or not and where it's located. And what we typically do when we're considering this is consider a duration of maybe a quarter of a second, about the average time that you would make a fixation when you're saccading around in the world. So that's it. And then if it's in overt search, where means you're allowed to make eye movements, then you start with an initial fixation. Stimulus stays on until you respond, right? You just keep looking around till you make a response. And so the goals or cost functions in this are to identify whether the target's present or absent. And if it is present, indicate where the target's located. And in the covert task, it's just to maximize accuracy, right? Just be as accurate as you can. And then in the overt search task, it's to minimize the number of fixations for a given error rate for finding the target, okay? And so that's it. So that's kind of the constraint, but it's still a pretty big range of behavior. Okay. And obviously you could, generalize this in the future, but that's kind of where we're at. So here's a generic framework for a theory of overt visual search. And I would argue that any sensible theory that biologically plausible will have these components, at least these components in it. So the input, of course, is the image, whatever is coming in. And also, we assume that whatever the organism has some prior knowledge about where the target might be located, right? So if you're looking on a search display, or if I tell you to look for something on this tabletop, right? There's a prior, it can be somewhere in there. And the prior can be whatever the person adopts. I don't necessarily mean by here the absolute true prior. It can be, it's whatever as a model, it might be what the organism's prior is. Or if you're doing searching in street scenes, like Antonio talked about, this prior might be, if you're looking for humans, might be more confined to where humans are likely to be. In addition, there's the observer, any sensible model observer is going to have some knowledge of its own visual capabilities. If I tell you that you're searching for a grain of sand on this tabletop, you know that you're not going to be able to see that grain of sand unless you get your eyes pretty close to it. So you're going to make one kind of eye movement. If I tell you, no, look for the glass on this tabletop, is there a glass there or not? Well, you're going to make one saccade in the middle, that's it, you're done. Right? So you know something about your own detectability. That's what I mean by deep prime, is how detectable it is across your visual field. So those are kind of the input, the sort of starting point. An image comes in, you encode it, then you compute some posterior beliefs based on that first eye movement of where you think the target might be. This doesn't have to be a Bayesian computation. I just mean some kind of posterior belief. And then based on that, you're going to make some decision. You either decide that there's no target there, you decide that there is a target and you indicate its location, or you don't know yet, and now you select an eye movement, where to move your eye next, and the process loops until you stop. Okay, so that's generic model of search, and we'll be interested in filling this in. That's overt search. Covert search, well, basically it's the same thing, except that, you don't get to make an eye movement. It's a kind of a one-shot. It's a one-shot deal on a saccade. And then this might be a little bit stranger. In covert search, of course, there's some external uncertainty about where the object might be located, right? There's just some external prior. But even if you know right where the target is, especially depending on where it is in the visual field, but even if you know it is, there's intrinsic location uncertainty in your visual system. And that means that even when you're searching for something where the target's known on every trial, it's still a search task. And so because of intrinsic position uncertainty, you don't know quite where your eye is pointing, and you don't know quite where, even if it's in the same physical location in the environment at other time, you don't know quite where it is on your retina, okay? So so that's the idea. So they're all search tasks, but we're going to start here, and then we're going to step back to here, and then we're going to step up to the general case. So that's what we're going to do. We're going to start with just an identification task where you, quote, know where the target can appear, okay? And what we're going to ask first is, well, what are the major factors or properties of natural backgrounds that affect our ability to identify a target? How do those factors actually affect the accuracy of detecting it? So that's important. And the reason for focusing on natural backgrounds is that's what the system evolved to do. That's what it's designed to do. So if you understand what it's doing and why it's doing it in natural backgrounds, you can translate that into laboratory stimuli, because presumably it's doing the same computations. It's not designed for, simple noise patterns or whatever clutter patterns in the laboratory. It's probably designed for handling the real world, and so if we got that, then we would take that model and then apply it to anything. Okay, so then, how does the variation processing across the visual field, right? You have high resolution neurophobia, worse in the periphery. How does that variation in resolution affect your ability to see things? That's going to be critical for search. And then how does intrinsic location uncertainty about where the target that's into the system, how does that affecting things? And then that intrinsic location uncertainty can sort of, if you remember Ruth's talk, can contribute to this idea of phase randomness in the periphery with texture and so on. Okay, so those are the questions, but before we get to those, I want to do a little tutorial. So this is the class part, and talk a little bit about target identification in noise, Gaussian noise. Okay, so some of you will already know this, but some of you won't, and we're going to sort of build on that, so I want to start with that. So we're going to talk about an ideal observer for identification in white noise, and then an ideal observer for identification in filtered white noise, just correlated Gaussian noise. So what's the task? And this is a nice way to start. So in this particular task, it's a single interval task, so we're going to present a stimulus for 1/4 of a second, right? Typical fixation duration. And if it's just white noise background, it just means every pixel has a random sample from a Gaussian distribution with some standard deviation. If the target's present, then whatever that target is, on the trials where the target's present, that's added in here. On the trials when there's no target there, then this would just be uniform noise. That's the task. And so just to describe the stimulus, so what does that mean? X here, bold X is like a vector, just means this XY coordinate pair, and we're just going to index them with I, that's all these points. So the background, if you have only a background only, it's got some mean luminance, that's L that you add to it, and then a random sample from a Gaussian distribution with some standard deviation sigma. And you have this independent sample at every location, that's background. If the target's added, the target has some target shape, that's T. this, and then it's scaled by some amplitude to set whatever the amplitude of that target is. Okay, that's it. If you do this in vector notation, here are all the intensities at all the image locations. We can just represent that by a big capital I. We can represent all that noise vector, all the noise things, by a capital bold N here, and the target by a vector describes the amplitude of the target at each location when it has an amplitude of 1. And then the image then, when it's background only, is the noise field plus the luminance, or the noise field plus the luminance plus the target. Okay, that's it. So what's the optimal? If you want to maximize your accuracy, what do? Probably obvious. This is pretty trivial. What you want to do is, when I get an image on a trial, I want to compute the probability that the target's present given that image. And I want to compute the probability the target's absent given that image. And then if the probability that's present is higher, I want to go with that. That's the best you can ever do in terms of being accurate. You can rewrite that a little bit using Bayes' rule, just using conditional probability. And what you want is the log likelihood ratio, which is the probability of the image given that it's present, divided by the probability of the image given that it's absent. If the log of that ratio is greater than the log of the ratio of the priors, then you want to say the target's present. That's just equivalent to this decision rule here. It's just rewriting it that way. In this particular case, if you just substitute into this, given your knowledge of these Gaussian random variables, and they're all statistically independent, so it's pretty easy to show that what you really want to do is just have a receptive field that has the shape of the target, and you just want to apply that receptive field to the image, that is, take the dot product or the inner product and get a response. And the response of that receptive field, if that's greater than the criterion, then you're going to say the target's present. If it's less than the criterion, you're going to say it's absent. Okay, so that's all there is to it. Just build a single receptive field that has the shape of the target and apply that. And that's the best you can ever do in this particular task. And how good is that performance? Well, there's this quantity d prime, it's just the signal to noise ratio, and it basically says how detectable the target would be. And what does that mean? A d prime of 1 would correspond in this task that we're in right now to about 70% correct. Okay, that's how accurate it would be. And so if you make that amplitude larger, this d prime, then the standard deviation, then your d prime will go up, and your percent correct will go up accordingly, and so on. That's completely understood. What we often do when we're doing psychophysics is we measure a thing that's called a threshold. It's just what's the amplitude of the target have to be in order for you to detect it with some level of accuracy. You can pick anything you want, that's fine, but a typical and a simple one in our case is to just suppose that what you want to be is 70% correct, and we'll just call that the threshold. Whatever amplitude of the target gets you 70% correct, then we're going to call that the threshold. And so that means the threshold, if you define your template here by taking the target shape and normalizing it by the energy of that target shape, then it turns out the threshold will just be the standard deviation of the response. And that's all this is saying. So that's the threshold, okay? All right, that's white noise. That's the best you can ever do. That describes the performance. Real images, of course, are not white. They're correlated over space in very complicated ways. But we can at least think about correlated white noise. And for example, this is what's called 1/f noise. It's basically filtered white noise, but it's filtered so that it has the power spectrum or the amplitude spectrum on average of natural scenes, at least natural scenes displayed on monitors, okay? And so what do you want to do here? Well, pretty simple. This thing was filtered in some way, so you just apply the inverse filter. That's called a whitening filter to this. And if it's just a background of noise, it'll just turn it back into white noise. Yeah, that's okay. So now we can turn it into the other problem. Only problem is that when the target's there and we apply this inverse filter, this whitening filter to the image, it's going to change the shape of the target too, right? It's going to whiten the target. But that's okay. what we're going to do is we'll just use the receptive field that's the Whiteant target. And now we're right back in the same problem we had. Performance is exactly just like before. So we just we whiten the image, have a whitened template, and then you just take the whitened receptive field, take the dot product with the whitened image. That gives you a response. If it's greater than criterion, you say, yep. And the D prime would be the same, and all that stuff. Okay? And this is called a whitened template matching observer. We'll just call this the template matching observer. Okay? And so you might ask, well, okay, that's a bunch of fancy stuff to do. Is it really necessary? How bad do you do here if you just ignore that and just stick with your receptive field that looks like your target? And it depends. So if you have sort of a narrow band, a sine wave target that doesn't have very many frequencies in it, would only stimulate a small population of cortical cells, then it doesn't make much difference. Maybe a 10% improvement, the threshold goes down, right? This is better, lower is better. 10% down over a template matching for detecting in the filtered noise. On the other hand, if you have a target that's got sharp edges and stuff like that, well, then you get maybe a 50% improvement by doing the whitening. So it really matters. So it depends on the situation. Yeah?
So whitening a sine wave just increases or decreases its amplitude?
It doesn't do much to it. It changes it a little. And the background, when you whiten the background, since it's a narrow band target, it doesn't really matter whether the spectrum is going like, doesn't matter what the shape is, you just have so much power that's in there, and whitening doesn't really change the power that's in the band that the target's in, so it just doesn't make any difference. Okay? So that's a useful thing to know. It means that you can actually you can kind of simplify the problem if you use narrow band targets. You don't have to quite worry about the effects of the structure of the background aren't quite as important. Okay? So that's one of the reasons that we're going to do some of that. Okay? So anyway, that's the tutorial part. So you know a little bit about template matching observers. That's the optimum in white noise. A whitened template matching observer, that's optimum in filtered noise, where there may be long-range correlations in the background. Okay, so now let's return and talk about our two questions. Start talking about covert search with no extrinsic uncertainty. And so what are the major factors and how do they affect accuracy? Well, these have been studied forever and ever. You heard some of it earlier from Felix and others. And here's just a little demo. There's been a lot of the whole history of psychophysics looking at these things. But here the same target has been added to both sides of these panels. And what you can see is if the luminance of the background is higher, then it's harder to detect the target. If you make the luminance the same on the two sides, but make the noise contrast, the contrast higher here, it gets harder to see the target. And if you keep the contrast the same on both sides, keep the luminance the same on both sides, but make the orientation and spatial frequency structure, pattern structure, more similar to the target, then it's more difficult than if it's got a different orientation and different frequency content. Okay? So that's the, well, these are big whopping effects. They've been studied for a long time. What we didn't know much about is how do those translate into the real world. That's fine. These are nice laboratory experiments. You can study them. That's great. And how do they interact? How do they combine in the real world? What do they do? So, and so what we have to study these kinds of questions, we've been using what we call a constrained sampling approach, and that's illustrated here. And the idea is, and this could be applied in other domains. So it could be applied in studying binocular vision or motion or other things. The logic is you take the dimensions that have already been studied in the literature that we know have a big effect on performance. And what you do is you then go into the real world and we have thousands of 14-bit resolution, high quality grayscale natural scenes, and we just divide them up into millions of background patches that are about the size of the target. And then what we do is we just simply bin those background patches along the dimensions that we know matter from the history of work and perception. And in particular, so this is just a histogram. So, where we've done this binning. So this is luminance along this axis, contrast on this axis, similarity on this axis. There's nothing to this, just a histogram. The color is only telling you how many patches are in there, so it's not interesting at that level. But the point is that every bin in this space is filled with patches, hundreds to thousands of patches that all have the same local luminance, all have the same RMS contrast, and all have the same cosine similarity between the amplitude spectrum of the target and the amplitude spectrum of the background. What does that mean? It's just a phase invariant measure of similarity, and by using cosine similarity, it's sort of independent of the other ones because it doesn't care what the luminance or the contrast is. It's just looking at the angle in the space between them. But it's a phase invariant. And the reason we use phase invariant is because we know that these masking effects that we see here, yeah, they depend a bit on phase, but largely they're phase invariant. So that's the reason for that measure, okay? So, why is this cool? Well, what's nice about it is that you could go in here and measure the threshold for every one of these bins, and now you'd have a pretty good idea about how these factors affect performance in real backgrounds. These ones that are well studied and well understood. Now, if you've got this many, one other thing I should mention, this histogram will depend on the target. All right? So these are two narrowband targets. This is a plaid that's got two narrowband peaks, and this is one that's got a single. But yeah.
So, but won't the contrast and luminance axes not depend on the target? It's only the similarity.
The similarity does, that's correct. But still the space as a whole, since similarity is on the thing,
Right.
yeah, correct. As long as these are the same size. They varied in size. Okay, so of course you say, well, gee, that's great, but it would take a couple lifetimes to measure all of these. It's not going to work, but no, wait. What's cool is that we know from the literature that the effects of these dimensions on performance are very smooth. No, luminance, in the laboratory, that's Weber's law. It's a straight line. So you don't have to measure everything in here. You can sparsely sample this space and now interpolate, and now you know how these factors affect detectability of any target you're interested with a very finite experiment. And so that's the power of the approach, is to give us some sense of how these factors work and how they combine and so on. To get a handle on what to expect and what's going on here, we can just go back to our simple template matching observer and say, well, what would that do? Yeah, these are correlated images, but we're using these narrowband targets, so we don't have to worry too much about that part of it. They're natural backgrounds, so they got more structure than that. But still, it's a good baseline to come in and see what would a template matching observer do on this entire space. And so you can just do that. Measure, just apply the receptive field template, this normalized template, to every patch in every bin. And these would be, for example, the histogram of the responses that you get from one of the bins in this space. And they're fairly Gaussian across the whole space. And that's because they're so constrained. So you may have heard of heavy tails, that if you just take if you take a receptive field like a simple cell receptive field and apply it to natural images, you get a sharp peak and heavy tails on it. But of course that's because you're mixing all backgrounds together. Here, we've constrained it to the similarity, luminance, and contrast. And in that case, then things are pretty Gaussian, which really just is saying that the reason for the heavy tails is it's a mixture of Gaussians, because these standard deviations are going to vary depending on the bin you're in. So anyway, you can write down the threshold is just the standard deviation, so we can just plot that for the whole space. And what's really cool is you get these really beautiful things come out. You got enough data here to pin it down. So this is a plot of all 2,000 bins for the two different targets. in the whole space here. So let me just walk you through this a little bit. So each panel here is for a different luminance. Oh, one other quick point to make is the range of luminance, contrast, and similarity that are in this histogram are picked to cover what you could present in the laboratory. There's actually the real range in real scenes is a little bigger than this. So this is constrained just to do things that what you can actually display. Okay, and so these are the different luminances. The contrasts are the color, the low contrast, the high contrast, and then the similarity, cosine similarity is on the bottom axis. And so, and the two different symbols, you won't be able to see them there, but you can on the slide if you get it. The two symbols are for the two different kinds of targets. And they fall on the same lines in this case. So what you see is this kind of beautiful thing, and the straight lines in here are this simple formula that says that the standard deviation, or the threshold of a template matching observer, is approximately proportional to the luminance times the contrast times the cosine similarity. So it's a separable product of those three dimensions. Yeah?
It's beautiful, but how many trials per subject? And how many?
This isn't trials. This is just scene statistics. This has nothing to do with, there's no behavior at all. This is just a template matching observer. Every stimulus was presented. So it's a lot of trials. This poor template matching observer was just completely worn out by the end of this measurement. So no, these are just scene statistics. It doesn't have to come out this way. There's no reason that cosine similarity, contrast, and luminance multiply in some product for what the standard deviation is. It's a scene statistics property. And it's a big nonlinear interactions here, right? Here's the effect of similarity. It's huge when the contrast and the luminance are high. It's minuscule. It's much smaller when the contrast is lower or when the luminance is lower, and so on. They're interacting in this multiplicative way. So this is separable, multidimensional Weber's law, if you want to know in psychophysics, for all three dimensions. So, okay, that's cool. Just one other quick thing to mention here. If you think about this thing here as sort of typical, narrowband targets is typical of what people think about, cortical receptive fields or wavelets for wavelet decompositions. This is describing the statistics of how those respond in the natural world as well. Okay, so you can think about it both as cells, or you can think about it as integrating all the information. Yeah.
Could you just clarify a bit? Is this separability dependent at all on the fact that these are natural scenes, or the kinds of targets you're picking and things like that? Or
the separability looks, so for example, we've been playing with, that's one thing we're doing right now, because we're kind of interested in taking this and making more realistic cortical neurons, for example. So one thing you can do is take, instead of just having a little gaborlet like this, we know that log gabor is a better description of cortical neurons. They're skewed, but they follow this same rule. And other ones we look at, there can be, especially too when you get to higher levels of similarity, there can be a little bit of deviation from this. There's some systematic changes, but the separability just is there everywhere. Okay. So how do humans compare? Well, so that we can do. We don't run them on, now you can ask about trials. But we just, this is an experiment where we ran, this is the average of several subjects. They all show the same thing, so I'm not plotting them here. And these are just along the cardinal axes. So what does that mean? That means in this particular plot, you go through the center of this thing and go along the luminance axis, freeze the other ones, go on the contrast axis, freeze those two, go on the similarity axis, just looking along those axes one at a time for the two different targets. That's what this is. So this is luminance down here for the two targets, contrast for the two targets, similarity for the two targets. The symbols are human thresholds. The little black dots are taken from the scene statistics of the template matching observer with one parameter in here. You've got to recognize you aren't as good as a template matching observer in this task. it's pretty darn good because it's a narrowband target. You've restricted things are pretty Gaussian. You're not that good. So this just has a single overall scale factor to bring the optimum down into registration with humans. But what you can see is that, but that single scale factor is applied to the whole thing. So these slopes and intercept differences and so on are all predicted from the scene statistics that we're seeing in humans. So they're acting, humans are acting in this experiment as if they also are proportional to this. Their proportionality constant just seems to be different. A little weak, not as good. Now this is not testing the separability prediction, right? It's a product. This is a recent thing we just did. These are the plots for the sine wave target from the other one, and then here's a separability test, and it looks pretty solid. So it looks like indeed humans' thresholds are consistent with separable multidimensional Weber's law across the three major masking dimensions that we know about. Okay, all right, okay, well that's cool. Looks like maybe we understand, we can predict anyway something about human detectability in natural scenes. But this is all under what we call, these are done under blocked conditions. And same with this template matching observer. It's in a fixed block, you just measure it, and the template matching observer can set its decision criterion separately for each individual block of trials that are run. Right? So that's not real world. In the real world, you don't know what the signal level is generally. You don't know what the backgrounds are. They're going to be randomly bouncing around. And not only that, you don't even know what the prior, and usually the prior probability that there's a target even there is usually low. In these experiments, it was like 50-50, half the times there was a target. So those are all unnatural things. And it turns out if you now take this simple template matching observer with a fixed criterion, and you just throw it in a situation where everything is randomly coming, signal levels and everything, it doesn't perform very well. And so you say, well, yes?
The target location is always known?
Yes, this is all known.
They always know this is where the target is?
Correct. Yep, absolutely.
And what is the single scale that brings the human and ideal observer?
It's just a proportionality factor on the product.
Is it 10, 100, 2?
In this particular case, I would. It's on the thresholds here. It would be a factor of 2 or 3. And so that's not going to work unless the template matching observer can estimate what the background properties are on every trial, and then dynamically, and taking the prior into account, what the prior probability of the target might be there, and dynamically pick an optimum criterion. So, and that would be the cognitive way of doing it. That's possible. But since you're always in the real world, always detecting things under a lot of uncertainty, you don't have to do that. You can do normalization. We've known for a long time that there are normalization mechanisms early in the visual system. And it turns out that if what you do is you have local mechanisms essentially that estimate the local luminance, the local contrast, and the local similarity of the target, and you just divide by that product. And you notice the order in which, since it's just a product, it doesn't even matter the order that you do these. You could do your luminance in the retina, you could do your similarity past V1, you could do your contrast in primary visual cortex, any order you want, or you can mix them together. Do some of your contrast in the retina, some luminance in the retina, whatever. Doesn't matter, but if you normalize by this, now you can adopt a single fixed criterion, and you can do just fine under these highly uncertain conditions. And I'm not going to go through the details here, but if that's what we would call a normalized template matching observer that's taking the scene statistics into account and doing normalization, then you get a lot better performance for the normalized, right? This is accuracy or hit rate, actually, versus the template matching observer. So it's a lot better performance. So that's big whopping effects. And then we've tested this in humans. Again, I'm not going to go through details here, where we just set up experiments where the contrast and the signal level and the contrast of the background are randomly varying both in natural images and in scenes. And again, and you can nicely show that humans behave like either they're dynamically adjusting their criterion on every trial, or they've got contrast normalization in their system. Because the template matching observer predicts this for low priors, for high prior, the target's there. For low priors, it predicts this. Humans fall right where they should. And so you can do that. The other really cool thing about this is it shows that what we always do in perception experiments, almost always, is we have equal probability of the two target being there or not. Turns out that's not as powerful for rejecting testing models. So you actually want to vary the prior probability, because that's when you get big differences in predictions. So that was kind of an eye-opener for us. Okay, all right, cool. So we're kind of rolling along here. It's a new explanation, actually, for why Weber's law exists. It's actually optimal, right? For those of you who know about photon noise, you'll know that the ideal observer, if you're just limited by photon noise, is the square root. So you don't expect Weber's law. That's non-optimal. But in the real world, photon noise is irrelevant. It's all the variation in the backgrounds that matters. In that case, Weber's law is the right thing. Weber scaling is the thing you want to do. Okay. So that's fine. But, a real cool feature about this paradigm, constrained sampling, I recommend it for other tasks as well, is that when you run this experiment on a human, you never have to, in fact, you don't want to ever present the same background twice. You're just sampling from one of these bins. You don't even do it with replacement, just without replacement. And that gives you lots more, in some sense, information. So you can still measure systematically the effects of the dimensions, but you never present the same background twice. So why is that cool? Well, it's because you got a whole bunch of trials where you've already controlled for the luminance, contrast, and similarity. You're inside one of these bins. And the trial-by-trial behavior of the person can then be analyzed to figure out what other factors that we don't know about, that haven't been studied in the past much, are controlling behavior. Okay? So that's kind of a good thing about it. We discovered one, and I'm not going to, again, take you through too much of this, but it's a cool one, and it's important. And it's a factor that had been picked up in the image quality metric literature. In engineering, there's a field where people try to develop image quality metrics. So people had noticed it, but had never studied it. They didn't, I think, quite know what to do with it. And so anyway, the other factors in here, this is just to demonstrate that there's something else going on. So every one of these rows are background patches from the same bin. This is a contrast of 0.2 and a similarity of 0.22. So these are 4 different bins, and all these backgrounds are from the same one. And so, but you can see, but they're ordered along this other factor. But what you can see is you can't see the target over here, worth a darn. And as you go further and further over to this side, it gets easier and easier to detect the target, right? It's kind of trivial. But a template matching observer performs exactly the same with these kind of backgrounds as it does with these. Well, what's going on there? Well, it's clear that's what's happened is these are kind of uniform texture regions. And then you've got regions where things are really dynamically changing underneath the target. And so that really makes part of the target visible, and part of it is invisible. It's masked by these other factors, but part of it just pops through. In some sense, you could think of it as it's occlusion, partial occlusion of the object by masking. And that's kind of what's going on. As I said, people had noticed this kind of phenomenon before, but hadn't studied it systematically. And so, it's pretty easy to see that this is an important factor. So we decided to take a look at it, and following Felix's suggestion, what we did is we started with simple stimuli and just simple noise. So let's just think about our white noise detector. But let's suppose that in fact the noise is not homogeneous under the target, but it can modulate. right? The properties of the background are varying under the target. And so here is a lion's head added to both sides. And of course, here it's uniform noise. Here it's high noise on one side, low noise on the other side. But the total noise energy power under the target, on top of the target, under the target, is the same in the two cases. So a template matching observer performs the same here as it does there if it's got a template of the lion's head, right? But obviously this is trivial because of the fact that you can see the features that aren't being masked as much. So that's the phenomenon. And so what do you want to do? What's the optimum thing to do in this case? Well, what you want to do is use, for those of you who are familiar with sort of Bayesian approaches, you want to use reliability weighting. What you want to do is you're looking for this particular target. Again, there's no uncertainty yet. And what you want to do is you want to normalize your template, what you're looking for, by whatever your estimate of the variance is at that location. And this is a reliability weighted template. And then you apply this template instead, and you can prove that that's the best you can do in this case for white noise, and that's the optimum decision rule. What's that actually look like? You got to be careful when you do this because you want this noise estimate here to be independent of the other noise estimate you get from the background over under the whole target. But anyway, this is sort of what the this is, you can just, I'm not going to take you through this obviously, but here's what the threshold is. The threshold is going to be the standard deviation of the noise divided by this thing here, which we call the partial masking factor. And what the partial masking factor is, it's just the norm of a normalized reliability weight, or it's normalized template. So the idea is you take your target shape, your template, you divide it by your estimate of the standard deviation at each pixel location, and you multiply it by the average standard deviation over the whole region. And this is our normalized template, not reliability weighted. And you take the norm of that thing, and that's the partial masking factor, and this is what threshold is. And then if you just realize in noise, this is going to be the luminance times the contrast, the standard deviation. So this is basically the formula for detection. Yes?
Sorry, I'm missing, how are you calculating the pixel-wise standard deviation?
Oh, hold on a second. Yep. And so the way we're going to, and this lambda is a parameter that indicates the local region. You're going to have to make an estimate there. Obviously, if you just knew it, you could write it down, but you have to make an estimate. And so the idea is that the dumbest way to do that is just have some little local computation of computing the standard deviation or computing the contrast in local regions within the receptive field. So like having subunits where you're computing local contrast in the receptive field region.
Sort of gone from the patch size effectively down to the small patch.
That's right, but you still have to consider the whole region because that's the target. And you still want to normalize over the whole region because that's the scene statistic. So you actually need normalization at multiple scales. But anyway, that's the idea. And so now you can sort of say, all right, well, we can test that and see how people do. So we ran this little experiment where this is, we modulated the noise from a 1-to-1 modulation, meaning it's uniform noise, up to 8-to-1, where you keep the total power under the target constant. And then we also modulated the frequency of the noise modulation, how many times it modulates per degree. And so there's like 13 conditions. The important point here is that for every one of these background conditions, all the combinations here, a template matching observer, they all perform exactly the same. And so then you just run a detection experiment and do it. So you get something that looks like this. These are so again, the prediction of a template matching observer would be the performance would be the same for all 13 conditions. There's 3 subjects. Here's the averages. Let's look at the average. Obviously, thresholds are not constant. You knew that was going to happen. Your thresholds are much lower. You're much more sensitive when there's a big modulation than you are when there's no modulation at this point or with a weak modulation. You can use this paradigm to estimate the spatial area over which people appear to be computing that local normalization. And it turns out it's about 0.21 degrees in this experiment. And if you estimate your local contrast over 0.21 degrees, then this is your predicted threshold, are these curves here. So it kind of captures what people are doing. They're behaving like they're doing reliability-weighted detection, but they're getting their estimate of the reliability over a certain window size. Okay, cool. That's white noise. Well, what about noise that's not white, right? Like real scenes. So we can go back to our 1/F noise, and we can look at all this, again, real quickly here. So we have a template match. So let's just consider where we've got contrast modulated 1/F noise, a little more naturalistic, right? Where the background contrast, but it's got 1/F power spectrum is changing. So here's uniform, here's modulated. There's a particular modulation ratio here. And you can apply a template matching observer, a Whiteant template matching observer, a reliability-weighted template matching observer that doesn't do any whitening, and finally you can combine them both. You can have a you can have a Whiteant reliability-weighted template matching observer. That's the optimum. This is the ideal that you can do in filtered noise. That's down here in these situations here, and wherever the purple one is up there for these different targets. And so what you can see is that that you get a benefit, again, for the sine wave target. Whitening doesn't help you much. This is, do reliability weighted template matching, right? That's what we were just talking about for this kind of stimulus. If it's just this sine wave, well, there's not much improvement. But if it's like a target with edges, then you get a big improvement like that, okay? If you just do whitening only, you don't get any benefit obviously from the reliability weighting. So I know that's a lot of data, information in there. Main point really to take here is that there's a big benefit from taking into account the modulations of the background under the target, and that if you're looking for kind of more broadband targets that have a lot of frequencies in them, what you really want to do, that's what this right here is, you whiten in spatial frequency, for those of you who know what that means, and you whiten in space. You can think of this as whitening in space by you're scaling the noise here to the same level. Okay. All right, so that's fine. What about natural backgrounds? And so now we're back. Now I can explain the axes here. So these are the same patches I was showing you before. But now what you can do for so now we're in a particular bin. We picked some bins that we have thousands of natural background patches in. And we can now bin those by their partial masking factor. And these are there's 5 percentile width, but there's 5, 25, right, 95. And again, these are the ones that are hard because the partial masking factor is small. Here the partial masking factor is big and you get better. And that's true for all of these, okay? So,
What is the partial masking factor again?
it's this guy right here. What you do is you take the template you're looking for, you divide it by your estimate of the standard deviation at each pixel location, right? You're getting it underneath every pixel. This is like a local contrast normalization or something like that. But it's scaled up. It's scaled by the average standard deviation over the whole thing. And you want to do that so that you're not doubly counting the background and contrast the background. And it's just the norm of this thing. And this is the factor. That's the thing that determines the effect of the partial occlusion by masking. So that's what we're binned along here. So now let's do the experiment.
So,
Yep.
so basically this doesn't care about the spatial profile of the modulations.
No, right, because it's just doing a local normalization. It doesn't really care what it is.
Right, so but is that also empirically the case?
We don't know that.
Okay.
I'm just going to show you what we know.
Okay. So,
But it's a good question. But what we do know is that the modulation frequency has an effect.
Right.
And so it does care in the sense that it's pooling over some area, and if it pooled over a smaller area, it could modulate its reliability more finely. But of course, then your estimates are not so good. Right? So what you want is to optimize the area over which you do it. There's a compromise about getting a good estimate locally and getting some benefit.
You say 5%, 25%, these are the,
This is just percentile bins of this thing. On the partial masking factor. So this is low partial masking factor. It's a homogeneous texture. We're in the same bin though, and so as you go over here, there's more modulation and it gets easier to detect the target. So what happens when you measure human thresholds across these for these different bins in the space, and you get just what we kind of expected you'd get. When the partial masking factor is high, your thresholds come down. It's a substantial factor. Of course, what we already knew from before is this is the easiest bin, this is the next hardest, the next hardest, and the next hardest of these 4 bins that we're measuring in here. So that's the vertical shifting. And then the partial masking effect is the effect that's running this way. If you now just run a simple template matching observer on these same patches, what you get what you should get, right? There's no effect of the partial masking factor. And these vertical shifts are just what we predicted before. This is the data I already showed you that we got for humans, these effects here. Now, if you take the partial masking factor into account with no free parameters, bang, you get this, and you get like 93% of the variance accounted for by this factor. And that pooling area and that factor were estimated just from the white noise experiment, frozen, and now you can generate predictions pretty well in natural scenes for human performance. So that suggests that maybe we have 4 separable factors that seem to be controlling detectability in natural backgrounds. Maybe these are the main ones. We don't know that yet, but if you take all of these together, this is big changes in performance. These are big effects. So it's a lot of the variance in overall human performance, and it's yet to be seen whether there's anything left. But these might be the major factors. Yeah.
This follows up on I think Roland's question. I mean, simply by looking at when you presented your mask in the noise, where it was also this partial.
Yeah.
This partial.
Partial masking. Yeah.
I mean, given that the partial masking was always vertical, the target was oriented horizontally. I think it seems obvious that it depends on the orientation of the target.
But that's the similarity effect that's still there. So this condition here has high similarity. This one down here has low similarity. That similarity is still captured over the whole region. So the model still gets that because it's looking under the target at how similar. So it's basically it's doing normalization at multiple scales. The normalization it does at the target level scale is picking up that similarity. And it's true, in this particular case, we're not using a local similarity there. And we haven't put that in, but at this point, it doesn't look like it's going to help us much.
I'm just thinking, just like in purely intuitive terms, you've got a 1D, suppose you've got a 1D target, and your contrast modulations are lined up with the stripes, so that the partial masking only reveals the bright.
You can make some phase thing that's really weird. Yes,
yeah,
right.
It's going to be. Whereas if you make sure that your modulations reveal just a narrow strip of dark to bright, dark to bright, dark to bright, this periodic signal, the total target's big, but you're just getting to see these periodic stripes. That's going to be presumably easier to detect than when you've got your modulations lined up with your targets.
But on average in natural scenes, you don't. It's always a mix. I mean, sure, that could happen. So if we want to call this a natural scene, we could line up with windows or something like that. But you know, it doesn't happen, and that might be some of the residual variance that's left down in there. Right? We haven't necessarily gotten every factor out of it, but these look to be big, important factors. And I like this paradigm in a way because it's a way of kind of studying, even with object detection, how occlusion works. You can use occlusion by masking, but you can actually use reliability-weighted models to do it, and so it makes kind of a nice platform for studying those phenomena. So, okay, so those are those two things. Those are four major factors. What about the variation in processing across the visual field? How does that affect target identification accuracy? Well, as Hans Vesely noted a long time ago, the sort of the big bottleneck is the optic nerve. Photoreceptors are a bottleneck, but ganglion cells fall off faster, so they're the real bottleneck. And in fact, if you like, the fall off in the retina counts for a lot of the fall off, most of the fall off that you see in the cortex, and that's a point that Vesely made. So one way to kind of account for things is just to measure the average fall off in ganglion cell density for the population of interest in the task you're in our case, it would be the P cells. And these are measurements from 6 human observers on average in the 4 directions, you know, horizontal, this way, that way, upwards, downwards, just getting density. And this is a little formula for the spacing that accounts for it. And you can generate retinas if you want, like this. So here would be the fovea, and this would be this basically represent the locations of the ganglion cell centers that have the average fall off in spacing between ganglion cells in the vertical, horizontal, and right direction, sort of interpolated. And what we know from lots of work is that this cortical magnification, which is close to that, by the way. So by the time you're in the superior direction, by the time you're 1 degree away from the center of your fovea, your resolution is down about half the midget ganglion cells. Okay, and so we know that that detectability tends to fall off inversely with the spacing between the ganglion cells. It's just a classic. A different way of expressing a classic result. But that rate of how fast the detectability of a target falls off, there's a target-dependent constant. So for some targets, it'll fall off rapidly, for other targets. And that's going to depend on a bunch of properties of the target. But there's a constant there that's unknown.
Is this an empirical phenomenological thing, or is it derived?
This is empirical. This is from anatomy, average of 6 human eyes.
Or the spacing function?
It's a spacing function. That's SP is the spacing function. And this is just, and we're only talking about central 16 degrees. Things get a little different out here, but this is just the spacing function.
Oh, that's anatomy, and they put anatomy in the equation on the...
This is the equation for the anatomy. Yeah. That's what this spacing function is right here. And then this is how sensitivity falls off, or threshold declines with spacing with a target-dependent constant. This is an approximation, but it's a pretty good one. And so you can kind of put that in here. We've got our 4 factors from natural scenes, and then we have a foveation factor that's got one parameter, and it's a target-dependent parameter, and it says the threshold is going to fall off like this. And so here you can kind of see, here's an image, here's the local luminances, here are the local contrasts, here are the local similarities for this target. Here's the partial masking factor. Here's the foveation, if you're fixating right here, factor. And then you come back, and now you have something that tells you about what the amplitude, this is sensitivity, 1 over threshold, but about what the threshold would be at every point in this scene for that background. And that's probably going to be pretty close. Okay, so what about intrinsic location uncertainty? Well, first of all, a little while ago, we stumbled across a nice closed form expression for the effects of uncertainty. So basically, position uncertainty means if I don't quite know, I'm looking over some region, but we're going to talk about looking over regions where my d prime is fairly constant. That's the whole idea. It's local. It's a local intrinsic uncertainty. And if I don't know where it is, I basically have to, what you can show is an optimum observer will actually apply the template, the feature it's looking for, over that whole region of uncertainty where it could be, and pick the max. You can also, there's other formulas. There's a true exact calculation. You can do the max rule. No matter what it is, it turns out that the d prime that you get with that level of uncertainty is described very nicely by this simple formula, where this is the d prime, or the signal-to-noise ratio, if you have no uncertainty, and then this is the effect of the intrinsic uncertainty. And the intrinsic uncertainty varies with retinal location. We've measured that in experiments. I won't go through it, but basically we know that the uncertainty increases the further you go into the periphery. And that's going to contribute to the kind of things Ruth was talking about, essentially the scrambling in some sense, is uncertainty about where things are located. Okay, so we've measured that. Oh, this is just showing you that it works. So you can simulate ideal observers for uncertainty, or uncertainty in natural backgrounds, or white noise, or orientation uncertainty, or whatever. Symbols are simulated, and then that simple formula are the lines, and it just always works.
What do you mean by uncertainty? You mean the subject does not know where the target is?
That's right.
Half or right half? Or
No, locally. It means that, so if I'm fixated here, and on every trial, this is only going to appear in one place, but I don't see it in one place. First of all, I don't know quite where my eye is, and secondly, my representation of position isn't all that great in the periphery. And so consequently, I could see a feature that looks like the target that's up here. It's not quite right where I thought it was going to be, but I'll learn in the experiment, my best guess is to say that it's there. If it looks like the target and it's near where I think it should be, go with it. You're going to be right. That's because we just don't have we have internal uncertainty. That's why detection with a target known location is still a visual search task. You're still looking around for that target. And so we measured that. Again, there's a target-dependent constant. That amount of uncertainty will depend somewhat on the target. EJ.
In terms of position uncertainty and this equation of applying over regions where d prime is constant, it looked like the partial masking estimate
Yeah.
varied quite a lot in space.
Yeah.
The regions where d prime are constant are
Yes, you're not wrong. This is just right. This is going to be an approximate. It's going to be an approximate thing. These position uncertainties in the fovea, for example, are quite small. In the periphery, the partial masking is probably integrations are over larger areas, and those more correspond better with the size of the uncertainty effect.
crucial parameter is how you scale over which you compute this partial masking.
What you can do in the real experiment, this is just a formula so we understand what's going to go on. And we're actually going to use it in white noise where it's not a problem. When it's in the real model, you don't do that. You just have the uncertainty and you actually apply it. You just add an intrinsic uncertainty into the search. So when it's really applied in a natural scene, this formula won't come in. Where the formula comes in is sort of in estimating the parameters of the human visual system, which we're going to do with white noise.
So a related question, if you think about the observer having access to many neurons or representations, not knowing which one to check,
Yeah.
is that expressed the same way mathematically as position uncertainty of the eye?
The same formula will work. It works for orientation. I haven't tried it for everything. Orientation uncertainty, that would be having different receptive fields with different orientation. It works fine for that. In other words, it's an accurate formula. It's accurate for position uncertainty. Spatial frequency uncertainty, I don't know.
Uncertainty about which neurons to look
Uncertainty, well, yeah, that, it ought to hold there too. Yeah, as long as, for example, which orientation on neuron to look at. Yeah, that's what I meant by it. I mean, we can talk more about that later. I don't have a precise answer for you here.
So can I say one quick thing?
Yeah.
Just since I think someone, you didn't talk about reverse correlation, is that right? You might just say, like, you can see this intrinsic uncertainty about the location. If you do reverse correlation sorts of experiments, you ask people to detect a symbol in noise, and when you look at the template that you infer from the reverse correlation, it looks like some blurry thing because they aren't sure exactly where the thing is supposed
That's
to be.
true too. And then we actually did a detailed experiment where we manipulated the range of uncertainty at different eccentricities and found out what level of physical, as the contrast of the target goes up, you sort of asymptote down to an asymptotic level of position uncertainty. You actually have to point where it is. And that comes out nicely in agreement with uncertainty increasing in proportion to the spacing between the ganglion cells. That's what the...
Why should there be a target dependence on this?
Well, because if I've got a great big giant target, I'm not going to get necessarily much effect on performance because I'm looking for this great big giant feature, and if I got a couple minutes of arc spread around, it's not going to make any difference. But if I'm looking for a fine feature, that plus or minus something is going to have a great big uncertainty effect. So it just depends. So anyway, I need to push along here quickly. But anyway, you can write down a full-blown model then that's got the D prime with no intrinsic uncertainty. That's the one we already have. I already went over this. Uncertainty has an effect on how D prime changes with the amplitude of the target, but there's another slight effect. And then you have the uncertainty effect. So you can plug all this in, and then there's a couple of parameters that are left. You can estimate those in a psychophysics experiment, what these target-dependent parameters are here, and this is overall scaling. And then you can move on to covert search. So that's probably a pretty good model of detectability, good first approximation of detectability in natural backgrounds. And now, once we go to covert search, what we really have is, this is before we have covert search, right? This is the model we were already talking about. These are the things, these are just scene statistics here. This is basically the anatomy except for one constant, and this is the anatomy except for one constant. What happens in covert search is, well, now we have some extrinsic prior over the possible locations of the target. And of course, the prior that that person actually has is also going to depend on what their intrinsic uncertainty is, so you combine those two. And then finally, you can allow an attenuation factor for some capacity limitations or things like that in here. And so what's the right decision rule in here? Well, there's lots of possible decision rules. The optimum would be you just compute the posterior probability that the target's present at every location. If that max posterior probability gets this likelihood normalized, yeah, this is the prior, gets above a constant, you'll say it's there. If you do say it's there, then you pick the location that's got the maximum, and like that. So we've started running some covert experiments just in noise to test this. And I'm not, again, we're really running out of time here, but I just mentioned a couple things about this task. So the task is that you've got a background of noise comes out, it's going to be briefly flashed, the target has some prior location of where it can be in the image. It's a simple target, sine wave target, so that we don't have to worry about whitening. And then what the subject does is they indicate whether the target's there, and if they think it's there, they indicate its location. And so that creates what you'd call in psychophysics, you get hits, false alarms, misses. False hits, that's where you basically got it right. There was a target there, but you pointed to the wrong location. And then you got correct rejections. And so you can, and so this model actually does okay in capturing that. It captures some of the trends in performance as a function of distance for fixation about where the target is, but there's certain things that it's failing on. For this, but this is pilot study, so I don't want to take too much out of it, but it's sort of looking like there may actually be a sort of a strange attentional effect that's coming in this experiment, but I don't want to, I could talk to Ruth about that, but I'm not going to talk in public about it yet because I don't believe it for sure yet. Okay, so that's covert search. We're going to do that more systematically, and then we're going to go to natural images because we should be able to make predictions there. And then overt search, well, now we have to add fixation selection in. So we've got all the stuff we already talked about, but now you have to have a stopping criterion. When do you quit searching? And you need to have rules for selecting fixations. So again, lots of details here, but I think hopefully this all kind of fits together coherently. If the target is always present in the scene, you're just looking for it, well, you just keep going until you get above some point. If the target might be absent, then you have a criterion for stopping, saying it's present or saying it's absent, and continuing. So it's not too much there. Here's the interesting part. How do you select the next fixation? Now we've got sort of an active vision task, right? What we're kind of have done is just consider lots of different possible hypotheses for what humans might do. Dumbest thing you could do is just randomly select from whatever your prior is about where the target might be. That's where you could have a posterior that you currently have. What's the posterior probability about where the target might be? And randomly pick from that distribution. That's another one. A so-called map is you pick the location that looks most like the target at this moment in time. Highest probability to be in the target. And that's a maximum a posteriori, that's a map search. That's not the optimum. You could get a little fancier. You could pick the maximum posterior if it gets bigger than some criterion, if you're really certain, but otherwise you just randomly sample from the posterior. And here are the approximately optimal ones. And so we've done some, it's related to Kalman filtering, but you can sort of derive sort of some optimal fixation rules. Maximum accuracy is you pick the location for your next fixation that, based on your current posterior probability over possible target locations, would give you the biggest increase in accuracy in identifying the location of the target. And that's not the location that's most likely to be the target, it's the location that's going to give you the most information. A closely related one is the location that's going to maximally reduce the expected entropy over the posterior distribution. That's a measure that will gain you the most information with a different information measure about where the target is. And these perform very similarly. And then you could have some kind of hybrid. Skip that. So what do these decision rules look like when applied to natural scenes? This is something we're, again, currently working on. And this is really cute. It turns out that what you want to do is this. This normalization comes in really interesting ways. And I'm not going to show you the proof of this. But if you have an image and you're fixating, what you can do is you've got your local luminance, contrast, similarity, and what you can do is you essentially have a map of that. I've already shown you that kind of map for a natural scene. You have your current posterior probability distribution over possible target locations that you got from your single fixation. That we already have seen how you compute that. Well, what you want to do is you actually want to normalize this thing, the posterior distribution, by this local luminance, contrast, and similarity, and get these normalized posteriors. And then once you have the normalized posteriors, what you want to do is you want to actually blur that whole image, that representation, by the detectability across the visual scene on a uniform background. It's your detectability map. And if you blur it with that guy, you'll end up with this bias map here, and you just want to pick the maximum of that. And that, in natural scenes, is the location that will maximally reduce the entropy over the posterior distribution of where the target might be. And so what's kind of cool about this is local normalization is a pretty common operation. We know normalization is Heger and Carandini and other people, and we have argued that's an operation that can occur throughout the nervous system and can be applied. And this is just a blurring by your own knowledge of your detectability and then picking a maximum. So, potentially these are things that you could do in the superior colliculus or somewhere else in the frontal eye fields. Anyway, so we haven't really got good search data yet with this, but that's the framework. And you can prove that. But we have done stuff with just searching in 1/f noise. Some of you may know this. And point is that these more complicated, more sophisticated fixation rules seem to work better. So here is a task where a target can appear randomly anywhere in 1/f noise that has the power spectrum of natural scenes. And you're starting in the middle, and you make eye movements until you find the target. Okay? Indicate where it is, and you have to indicate where it is. And then this is just the plots down here of the distribution of eye landing points across all the search trials in the experiment. And what I mean by that is we had different noise levels of noise background and stuff like that in the experiment. So there's a lot of trials. And what you see something like this. So this is the display. That's the boundary of the display. And then this is the distribution of fixation locations just across all the trials in 2 humans. And what you can see is a couple just interesting facts. Humans don't fixate very near the edge. That's common sense, right? Because if I fixate right near the edge of this display, you won't do that. Why won't you do that? You won't fixate that because you're going to waste good fovea over here outside the display. You're just not going to, you won't do that. And you don't. The other thing that's interesting is you actually spend more time fixating in the upper and lower visual field and not on the sides. Your acuity is best on the sides. So what you tend to do is know that your acuity is best on the sides. So you got to spend more time in the top and bottom because that's your weak spot. And if you either pick fixations that maximize your accuracy or maximally reduce the entropy of the posterior distribution, you get these kind of donut distributions with hotspots in the top and bottom. But if you say always fixate the location that most looks like the target, you get a completely different pattern. You actually make more fixations laterally and less into the top and bottom. And you go near the edge of the display, which you don't do so much with these. Because, again, these are trying to maximize gain in information, so one thing you don't want to do is waste fovea. And so humans show some of those properties. Yes?
Does this data exclude saccades to the target itself, in the sense that, so do people generally respond without saccading directly onto the target? I can imagine that like their final saccade is on the target and then they make the response.
What we do is the way the task works is they go, and as soon as they see the target, they hit a button.
Right.
Then they fixate the target and press it again. So we don't require them because otherwise they're going to slow down. Oh, that's the target. I'm going to stop there, and then you get all these fixations that are right. Yeah, we don't want to. Yeah. And then with Vivian, is she here? There she is. Vivian and Carl ran a similar study with a different configuration of target positions. They're not uniformly distributed, and they ran it in scotopic conditions. And under scotopic conditions, you're now using your rods, so the whole distribution of how sensitive you're going to be at different locations shifts. And so if you're optimal, what will now happen is you won't get a donut distribution anymore. This is what an optimum does, who's got under scotopic conditions with this kind of configuration of possible target locations. And you fixate, again, you tend to fixate a little bit above because you're most sensitive below, and you want to go up there a ways because that's where your good sensitivity is because it's under rod vision. And humans are similar. Again, you don't fixate near the edge of the display much or anything, and you have this. This is under photopic conditions with this kind of target. You'll notice you also don't get a donut distribution, right? It doesn't look like this. Why not? Why doesn't it look like that? Well, I know humans do this. Why doesn't it look like that? Well, oh, come on. Oh. It's because you don't gain as much information going out here because of the sparseness of the possible locations. So the ideal is to fixate closer, and this okay? And not exactly the same, but qualitatively in the right direction. Okay, so that's it. That's a attempt at starting a formal theory of search in general backgrounds. We used natural systems analysis to develop a theory of search from first principles. It's based directly on natural scene statistics. We found there were 4 separable factors that seem to predict identification accuracy in the fovea for additive targets. We can talk about that. That's a limitation. 2 biological factors, sampling density of ganglion cells, which corresponds a lot to sampling density of the cortex, and intrinsic position uncertainty, which grows with eccentricity. And so we're trying to build a general theory that's built on this foundation by including optimal and suboptimal hypotheses for about how things are pooled and how fixations are selected. And obviously it's a work in progress. Thanks.